Hello, I'm Shigeyuki Kakizawa from Japan. And today I'd like to talk about the serum and albumin-free media for mycoplasmas, including the minimal synthetic cell, JCBI, Sin3, Sin3b, and Sin1. And first, I'd like to explain why we need serum-free media. So since many mycoplasma species are unable to synthesize lipids, so they require serum for cultivation. However, serum has some disadvantages. For example, lot-to-lot variation, difficulty in proteomics due to high albumin content, like that. Here is the SDS page here of media, and the high cost, and the animal welfare concerns. So if serum could be removed from media, there would be various advantages, making it useful in many purposes. Here is an example of a previously reported serum-free media in mycoplasma. So this is for the mycoplasma pneumoniae, so the pathogen for the pneumonia. But this is a very important and famous work by Spanish group. And they did a lot of the metabolic pathway analysis using the mycoplasma pneumoniae. And then during these procedures, they successfully established a defined media for mycoplasma. Here is the content. And then they include several kinds of, 3 kinds of lipids. And then BSA, it is a lipid carrier. So the defined media includes the cholesterol, palmitic acid, oleic acid, and BSA. And here is another example of previously reported serum-free media. These are also from the Spanish group. And then this is the Mycoplasma pneumoniae and other strains, and the Mycoplasma hypopneumonia. And these media includes the lipid and BSA. So according to these good examples, previous work, we aim to develop serum and albumin-free media for mycoplasmas. And we first use the minimal cell JCBI Sin3b as first our initial target. So we tested various media and concentrations, including the lipid carrier, PBA, polyvinyl alcohol, and the cyclodextrin, and the other component, including the detergent twin, glycerol, sugars, acetate, and so on. Today, I will skip all of these trials, but finally, we successfully developed serum and albumin-free media for Sin3b. And then we found that the combination of 3 to 5 types of lipids with cyclodextrin and PVA was really important. So we added 3 types of lipids, 3 to 5 types of lipid like that, plus 2 lipid carriers, cyclodextrin and PVA. And we named these media as LPC media, which stands for lipid, PVA, and cyclodextrin. We made several versions of LPC media with various concentration of lipid and lipid carriers, like that. And then there is no visualized band in the new LPC media, like that. So almost no band. But still there is a small peptide like that, but there is no obvious the larger band compared to the FBS. Here is the growth rate of Sin3 and Sin3b. And the red line shows the growth curve with SP4 media with fetal bovine serum. So the growth speed in the new media was slightly slower than in SP4, but still acceptable, especially in Sin3b. And the media without lipid carriers, so that means only lipids, were not effective at all. And we did the RNA-seq and the proteomics analysis of Sin3b. And here is a result, SP4 versus the lipid non-serum media, LPC4. And here is the RNA-seq. and the proteomics. So after, so the expression level of most genes remained largely unchanged at both RNA and protein levels. So the newly developed serum-free media could potentially serve as a viable alternative to the original media. And also we tested number of colonies using the solid medium, using our LPC media, and it's really good. And we did tested the recovery from frozen storage. LPC 1, 2, 3 is not good, but 4 and 5 are really good. So the minimal cell Sin3b showed the comparable result in colony formation and the recovery from cryopreservation using the LPC media. Then we tested the other mycoplasma. The first choice is Mycoplasma gallisepticum. It is a chicken pathogen. And SP4 is not good, but R2 and Felis and the Frey media, our LPC content is really good to support the growth of the gallisepticum. And then we tested the Synoviae. It is also chicken pathogen, and the pneumonia, the human lung disease pathogen, and they are really good. And we tested the spiroplasma, 2 species. And in some media, they showed a good growth. And lastly, here is the price of media. Especially in the SP4, the content of FBS is higher than other media and really expensive. So our new media is less expensive than original media. And recently, we found that 2 additional lipids are really good for cultivation of Sin3. Firstly, we choose the 3 lipid plus lipid carriers, but additionally, we added sphingomyelin and the phosphatidylcholine. And after adding these 2 additional lipids, the Sin3b shows really good growth like that, almost better than the SP4. So Sin3b showed better growth in the new media, and we are keeping to improve the media now. And this is some note for the minimal cell. Sorry about that. This is a really small letters, like characters like that. But we did various concentration of lipid carriers, cyclodextrin and PVA. And then there is the optimal concentration range for lipid and lipid carriers. And most components were found to be harmful when added in excess. And also here is another tips, but a high concentration of glycerol was found to be harmful to Sin3b, and preparing the glycerol stock for freezing was not suitable. Here is a summary of this media. Mycoplasma is a pathogen of diseases, and many media have several problems due to the serum. And we developed the serum and albumin-free media. And it has several advantages like that: cost-effective, fewer animal welfare concerns, no lot-to-lot variability, and then no visible proteins. We hope it will be widely used in the future. And I acknowledge all of these people, our team, and the AIST team, and also John Glass, Yo Suzuki, and Kim Wise. That's it. Thank you very much.
Hi, this is James Dobbins Speck from University of Alabama in Birmingham. I did quite a bit of work with that, using that 2009 formulation with their serum-free media. And one of the tricks was you had to solubilize the lipids in ethanol, hot ethanol, and then inject that into about 2 grams of serum albumin, and then put that in the media and get a stable suspension, not a solution, for about, and it would stay stable for about 2 weeks. So if I'm reading this right, you're using the cyclodextrin as a carrier instead of albumin, right?
Yes.
If I remember the structure right, that's a circular glucose containing molecule with 6 glucoses in it.
Yes.
So really my question is, do you have any tricks like the ethanol trick, right? That you would take, and are you solubilizing your lipids in ethanol before you put them with the cyclodextrin? Because
yes,
almost all those lipids are non-soluble in aqueous.
Yes, we prepare the first lipid stock using the ethanol, and then added into the media. But we did not observe some effect by ethanol because it's really tiny amount of ethanol in the final media. So we are not sure that cyclodextrin or PVA has a kind of the effect for solubility of the lipid or not.
Thank you.
Thank you.
Shige, did you have to try different cyclodextrins?
Unfortunately not. We choose just one, beta methyl cyclodextrin.
And how close are you to publishing these results?
We are now writing a paper, and
Good.
hopefully in 1 month or something.
Mhm.
Yeah.
Have you looked at James Sayens's papers doing similar work?
Yeah, but I imagine that they are using BSA, maybe.
I think they are. Yes, they are.
Yeah,
Mhm.
yeah. So the big difference is removing BSA and adding the two lipid carriers. And we tested various concentration of BSA, but maybe according to our experiments, the BSA is not so good for cultivation of Syn3.
Okay. Do you still get the same sort of fried egg looking colonies on agar plates?
Yes. Fried egg colony, yeah. In all our new media.
Well, this could be a huge thing for all of mycoplasmology, I would think.
I hope so. Yeah, thank you very much.
Really nice, Shige, and I really look forward to seeing this paper. Okay, so we're
John, can I make a question?
Oh, yes, please.
Hi, this is Daniela Bittencourt. Hi, Shige. Thank you very much for your talk. Just one minor question. How long is the best incubation time for C3B using the better solution components that you found in your new media?
You mean the incubation time?
Yes, like when you get when the perfect growth, like log phase.
Yeah, actually the growth speed or log phase is depending on the dilution rate of, and also the how active in the cultivation, cultivated cell. So
yes, but have you tested if you use the same incubation amount and the same volume as before and in the medium that to get the best result? Would there be differences for how long you need to incubate them to get?
Usually around 24 hours, the growth is really good. Yeah, but we can increase the inoculum. And then if we increase inoculum, for example, 1% or 2% of media, So we can see really good cultivated cells around 18 hours. But
Nice.
usually, if we put, for example, 0.1% of inoculum cell into the new media, we need more than 1 day, 1.5 days or 2 days.
Okay, I
Yeah.
understand.
Mm.
Okay, so yeah, so it's good because the media has the right amount of nutrients necessary to grow in 18 hours if it keeps going well. So this is nice. Thank you, Shigeyuki. Thank you.
Yeah. Thank you.
Shige, I was in thinking back through your talk, you showed a acrylamide gel with proteins in your media. I would have expected to see some proteins from the yeast extract and the Mycoplasma base broth. Where were they?
Yes, in our content there is still yeast extract and PPL. Yeah, but there is no obvious protein band in our media because maybe the content is really tiny or degraded. And
Okay.
that's why there is obvious band. Yeah.
And then also you used and I probably missed this, there was the abbreviation PVA. What is PVA?
It's a polyvinyl alcohol.
Okay.
It's kind of the sticky material to seal the papers or like that.
And so, okay, so this is something that, what led you to put PVA in the media?
Yeah, this is a very important point. Previously, for the mammalian cell cultivation, it was reported that the PVA can replace BSA and work for the kind of the carrier lipid, and also the some paper report that PVA can have function for reduce the harmful effect from lipids. So the inspire of the human cell or the mouse cell, the cultivation, we tested the PVA to mycoplasma.
Okay. Other questions? Kim, got anything?
Yeah, I was just thinking, Shige, of course, really nice work. I can really appreciate what that all means. But I wonder, over long-term passage in that medium, do you notice any change, presumably an increase in the growth rate? And have you considered maybe sequencing the genomes after long-term passage to see if there's any changes, mutational changes in some of the pathways that might affect the lipid synthesis in particular?
Yeah, thank you very much. We are really interested in that, but we did not do that. So maybe we hope that after the laboratory evolution using the serum-free media, maybe since we can, genome will be changed, but we didn't do that so far.
Yeah.
Thank you.
Okay.